Good afternoon to everybody. I am Josephine Mini from the Department of Botany, Women's Christian College, Nagarkoil. The topic that I have selected for my presentation is micropropagation. Micropropagation refers to the in vitro multiplication and or regeneration of plant material under aseptic and controlled environmental conditions to produce thousands or millions of plants for transfer to the field. The German botanist Haberland is considered as the father of tissue culture, who conceived the concept of cell culture in 1902, and also he has conceived the concept of totipotency. In India, commercial tissue culture was born in the year 1987 when A.V. Thomas and Company Kerala established their first production unit in Cochin for clonal propagation of superior genotypes of selected cardamom plants. When we talk about micropropagation or tissue culture, the two terms should be very carefully maintained, that is the aseptic condition and controlled environmental condition. To maintain the aseptic condition, certain sterilization techniques are used. Dry heat sterilization is used to sterilize the glasswares, and it can be done in a hot air oven. Flame sterilization is used to sterilize the needles and forceps while inoculation, and this is done under a flame or spirit lamp itself. Autoclaving is the procedure for sterilizing the tissue culture media. For autoclaving, the instrument autoclave is used. It works under the same principle that of a pressure cooker that we normally use at home. Up to the sterilization of 500 ml of media, the temperature can be maintained at 121 degrees centigrade for 15 minutes. Filter sterilization can be done for amino acids and vitamins because this can be destroyed by heating. And also for the culture of spores, this filter sterilization can be used. Wiping with 70% alcohol is done for all the equipments which we do micropropagation. Even before inoculation, our hands should be sterilized with 70% alcohol. Surface sterilization is done for the S plant which we are going to inoculate. The S plant is the small plant tissue which is used for multiplication and the production of large number of plants. Controlled environmental condition. The temperature should be maintained between 20 to 30 degrees centigrade. Normally the temperature maintained is 24 plus or minus 2 degrees centigrade. Then the light is 10,000 lux. For normal plant growth, the media used is soil. It grows in soil. But for tissue culture, there are certain media available: Murashig and Skook media, Lensmer and Skook media, Gambok medium, etc. The commonly used media is the Murashig and Skook medium, which was originally formulated by Murashig and Skook in 1962 to optimize tobacco callus. This media will contain certain micronutrients, macronutrients, vitamins. Agar is used for the solidification of the media, then sucrose and even plant growth regulators. There are certain steps involved in micropropagation. First, we have to select the stock plant and bring the stock plant from the field level, and it should be grown under controlled condition. It can be grown in our own garden or under a greenhouse. From that plant, the S plant is excised off, and this S plant is transferred to the medium for the multiplication of shoot or rapid somatic embryo formation. The transfer of S plant to the medium is called as inoculation. This inoculation should be done under a laminar airflow chamber where high efficiency particulate air filter is present in laminar airflow chamber, which brings ultra clean air from inside towards the outside, so that it prevents the entry of microorganisms. After inoculation, it is maintained in the culture room so that multiplication of shoot or rapid somatic embryo formation takes place. The shoot formation directly is called as direct organogenesis, which can be obtained from bud culture, and rapid somatic embryo will be formed from the callus, which will be obtained from a group of tissue such as a small piece of leaf or stem portion, and from the callus the somatic embryo forms, and from the somatic embryo shoot system arises. So it is called as indirect organogenesis. Then the shoot formed is then transferred to the rooting media. For rooting media, their plant growth regulator auxin can be used. The multiple shoot will be arranged, and from that individual shoots should be transferred to the rooting media. After the root arises, the plantlets can be transferred into the sterilized soil for hardening under greenhouse environment, because these plants are grown under controlled condition. Directly it cannot survive in the field. The plant organs will be very delicate, so that for few days it should be maintained under greenhouse in sterilized soil. Based on the S-plant used, there are different types of cultures such as seed culture, embryo culture, meristem culture, bud culture, callus culture, anther culture and protoplast culture, where I will discuss only few of them. This is the embryo culture. Immature seeds are selected and then the seeds are sterilized. The sterilized seeds are then peeled off to remove the glums. Then from the glums removed seeds, the seeds were cut open and the embryo is excised off, and this embryo is placed under the growth media, and from the embryo the plantlet arises. Then this plant can be transferred to the sterilized soil and transferred to greenhouse and then to the field level. Here from the embryo plantlet arises directly, and so this process is called as direct organogenesis. Here comes the bud culture. Here the nodal part is used as the S-plant. Usually from the node an axillary bud develops and forms the shoot. Here we can see the bud development and shoot arises. At the base of the stem we can find the formation of callus. Callus is the mass of parenchymatous undifferentiated cells. From this callus somatic embryo will be formed, and there are number of shoots arises from the somatic embryo. Each shoot will be individually transferred to the rooting media, and again from the rooting media after the root arises it is transferred to the soil. Next is the anther culture. Haploid plants can be produced by anther culture and also pollen culture. For anther culture, anthers can be directly inoculated, and from the anther embryo is formed, and from the embryos shoot develops. And for pollen culture, from the anther the pollens are taken and the pollens are sterilized and placed under culture media where the embryo forms, and from the embryo the plant develops. Some of the applications and merits of micropropagation. A small amount of plant tissue is sufficient for the production of large number of plants. Then large number of plants can be maintained in small spaces. It also helps to save the endangered species and store the germplasm of the particular species. Disease-free plants can be produced through meristem culture. The proliferation of in vitro seeds can be done at any time of the year. We don't want to wait for a favorable season to come. Small-sized propagules can be stored and transported easily. Then genetic uniformity can be maintained, because always the media and the environmental condition, everything remains the same, so that the genetic uniformity can be maintained. Synthetic seeds can be produced through somatic embryogenesis. Some of the disadvantages: contamination of the culture takes place if we don't maintain proper aseptic conditions. Even bacterial and fungal contamination occurs. Breathing of the media. Some of the plants will contain secondary metabolites such as phenols, which will be exudated into the media, and it interfere in the growth of the tissue, so that certain steps should be taken, such as adding of ascorbic acid or citric acid to the media. Vitrification. When we do some large subcultures, the water content of the plant increases and the leaves become translucent. This can be avoided by the addition of agar. Even addition of more agar leads to the interfere with the growth of the tissue. then expensive equipments and sophisticated facilities and trained manpower is needed, so that this expensive equipments, sophisticated facilities and trained manpower limits the use of micropropagation. Thank you.